The axolotl is an amphibious creature originating from Mexico. It has been a model species for over 150 years. The genome has 32 billion base pairs compared to the mere 3 billion in humans. That is over 10 times as big. Although this creature has a vast genome, that does not dictate the full complexity of the axolotl itself. Here, we see two axolotls battling it out for the delectable worm. The fight turns vicious, and an arm is lost. After an amputation, it has a remarkable capability to regenerate. They can grow back a nearly perfect replica of almost any body part, including their spinal cord and up to half of their brain. Scientists are trying to determine how the regeneration in axolotls work in order to potentially use that mechanism in humans.
Mwahaha! I have done it. I've edited the CRISPR-Cas9 protein with axolotl DNA. It'll change the arrangement of my chromosomes, giving me the power to regenerate, and with it, I will be unstoppable.
Wait! You don't understand, sir. That's not how that works at all. The CRISPR-Cas9 works as a transcriptional activator.
A CRISPR RNA is attached to a Cas9 enzyme. The CRISPR RNA is modified to recognize a specific sequence and tells the Cas9 where to cut. After the chromosome is cut, the cell tries to repair itself. This can be done either by simply gluing the pieces back together, introducing a deletion mutation, or the cell will add a couple random base pairs between the cut, introducing a random mutation, both often rendering the gene non-functional. In order to create an intentional mutation, donor DNA can be added to act as a template to fix the damaged chromosome. This process can hypothetically be the solution to adding the axolotl's regenerative properties to the human body. However, much is still unknown about CRISPR-Cas9 and the axolotl genome to be certain quite yet.
Research is still being done, sir, and there hasn't been any human studies done yet to see if regeneration properties are possible in humans.
Silence! Nothing you can say can stop me!
Many months later.
You have several abnormal genetic mutations. And no, they don't cause superpowers.
Help, my axolotl doesn't have an arm.
Alright, let me just consult my geneticist friend real quick and I'll see what we can do for you.
Hello?
Hi there. I have a client who has an axolotl with a missing arm. What can I do for her?
Oh yeah, no worries. With an axolotl, that arm will grow back.
Oh wow, that's really cool. How does that work?
So when regeneration is happening, cells nearby turn into an unspecialized state, like stem cells, and form what's called a blastema. This means the cells can become in one of many different kinds of cells. These dedifferentiated cells in the blastema then grow and multiply, eventually regaining their identity as fully developed bone or skin cells. That information is in the chemical tags on the epigenome that can tell the cell where they are.
Wow, so is there any way this can be replicated for human cells?
Researchers haven't found a human counterpart gene yet. However, the enormous genome of the axolotl has yet to be fully sequenced.
Sequencing? What's that?
So sequencing is the process of finding the order of bases in DNA strands. First, a single DNA strand is divided into smaller pieces and inserted into plasmid DNA, which is then put into bacterial cells. And as the bacterial cells multiply, so does the DNA. This duplicated DNA is then isolated and heated to separate into two separate strands. And added to these strands are free DNA bases A, C, G, and T, DNA polymerase, DNA primer, and modified bases labeled with colored fluorescent tags, also known as terminator bases. After the temperature is lowered, DNA primers begin to bind to the plasmid DNA, followed by the DNA polymerase, which proceeds to add free bases to the strand until a terminator base is added, which does not allow any more bases to bind. After this, it is heated again to separate the small fragment from the template strand. This process of heating and cooling is then repeated many times to produce numerous different fragments of varying lengths, which is determined when the terminator base is added. These fragments are then separated by a gel with an electrical charge at the opposite end. DNA is negatively charged, so they are attracted to this positive charge. The shorter fragments move more easily through the gel than the larger parts, resulting in arrangement by size down the gel. The terminator base is then illuminated to display the fluorescent tag associated with this base. A is green, C is blue, G is yellow, and T is red. These colors are then recorded. With this line of colors, we get the sequence of our DNA. This is only for that small fragment, however. This entire process needs to be repeated countless times to get all the fragments sequenced. Then, piece those fragments back together like a puzzle. So you can see this process is quite laborious with the vast 32 billion base pairs of the axolotl.
Thank you for this consultation.
Oh yeah, anytime.
So after my consultation, your axolotl will just need to stay with me for about a week and that'll cost you about $2,500.